When cytotoxic T lymphocytes encounter a cancerous or pathogen-infected target cell, they form a specialized contact site called the immunological synapse, where they secrete the deadly contents of cytotoxic granules. But, as Jens Rettig from Saarland University explains, cytotoxic granule membrane components must then be retrieved by endocytosis, so that the lymphocyte can quickly replenish its granule pool and kill additional target cells.
The flower protein was reported by Hugo Bellon's lab in Drosophila neuromuscular junction to be a calcium channel which is responsible for mediating the calcium dependence of endocytosis of synaptic vesicles. And we thought, well, let's have a look whether that protein plays a role in cytotoxic granule endocytosis as well.
Rettig and colleagues, including first author Xin-Fang Chang and co-senior author Veit Flacherzi, found that the mouse homologue of flower is expressed in cytotoxic T lymphocytes. The researchers then generated flower knockout mice and examined how the loss of this protein affected cytotoxic granule dynamics.
We did a series of controls to really make sure that exocytosis, which is of course preceding endocytosis, is not affected at all. So we did a so-called degranulation assay and we didn't observe any difference there. And then we looked also in total internal reflection fluorescence microscopy, where we looked at the fusion of individual granules and also didn't see any quantitative difference in the number of cytotoxic granules being released at the IS.
To monitor whether endocytosis is impaired in the absence of flower, Chang et al. Used a fluorescent marker of cytotoxic granules and an antibody that recognizes the marker's fluorophore when it's exposed at the cell surface.
With wild-type cells, we observed a certain rate of recycling of these cytotoxic granule membrane components, and in the flower knockout, we found that this endocytosis was almost entirely blocked, so the fluorescence remained at the IS over a time frame of at least 15 minutes. We performed so-called correlative light and electron microscopies, and we didn't find any invaginations within the plasma membrane, so we think that the block of endocytosis occurs at a very early stage.
This block in endocytosis reduced the ability of flower-deficient T cells to act as serial killers capable of killing multiple target cells. Endocytosis could be rescued by expressing exogenous flower or by raising extracellular calcium levels.
If the flower protein is somehow working as a calcium channel and we provide the calcium through other sources, we would expect a rescue, but although we really tried very hard, we were never able, also with electrophysiology, to measure any calcium current through these channels. So yes, extracellular calcium certainly is rescuing it, but whether it is substituting for the lacking calcium channel function of flower, we don't know at the moment.
Regardless of whether or not flower acts as a calcium channel, Chang et al. Turned their attention to the protein's localization.
Initially, Hugo Bellon was proposing that it's localized on synaptic vesicles, and of course our expectation was that it is then localized on cytotoxic granules in T cells, but it certainly is not. It's not localized on cytotoxic granules, it's not localized on any kind of endosomes, but it is localized on vesicles. And at some time point, a minor fraction is also localized in the plasma membrane. We do not know anything about the identity of these flower-containing vesicles, but we know that these vesicles move to the immunological synapse before the cytotoxic granules move to the immunological synapse. So they are certainly acting there at a very early step.
Chang et al. Found that flour binds to the adapter protein AP2, a key component of the endocytic machinery. A flour mutant unable to bind to AP2 showed an increased localization at the plasma membrane, but couldn't rescue the endocytic block in flour-deficient T cells.
So it might be that the function of flour is not in the plasma membrane, it is in the vesicle, or the other alternative is that it really needs to bind to AP2 in order to trigger endocytosis. And this is actually our favorite hypothesis, that it really builds a nucleus for AP2 to bind, and we know that AP2 binding to proteins which are going to be endocytosed is a very early trigger for endocytosis.
So what do Rettig and colleagues want to investigate next?
Well, the key question for us to look at is what is the identity of these flour-containing vesicles? What other components are on there? It might be, for example, that they contain other endocytic proteins like clathrin, which is also required at an early stage of endocytosis.
For now, though, you can learn more about how cytotoxic granule endocytosis depends on the flour protein in the paper by Chang et al., published in the February 5th, 2018 issue of the Journal of Cell Biology.
You